I'm going to talk about association analysis of quantitative traits. I will start with a short introduction and then we will discuss different aspects, different ways to measure association, and then finally we will discuss what is the specific of association analysis of genetic data. So here I have 4 data sets presented in panels A, B, C and D. In each panel I demonstrate the relation between two quantitative variables x and y. So each dot represents measurements from a single sample. You can think of this, for example, x is the weight of a person and y is height of a person, and well, dot corresponds to a particular person. So if I want to answer the question where the association is stronger, it's probably quite visible that at the panel A the association is strongest, and then in panel D it is a little bit less strong, and then B and C are relatively the same, so maybe in C the association is slightly stronger. I'm going to ask the same question about these 3 different data sets. So would you agree that it indeed looks like that association in panel A is stronger than in panel B, and stronger than in panel C? To give quantitative answer, we need to introduce a way to characterize association between 2 variables. What about using coefficient of regression of y onto x? Does everybody expect that indeed regression coefficient for the panel A data would be greater than for panel B than for panel C? So let us start with defining this measure of association, linear regression, and for that we need to define the linear regression model in which let y will be an outcome and x would be a predictor variable, or you could call y a dependent and x an independent variable. So let y and x with subscript I are the outcome and predictor values for a particular sample. So in linear regression we would assume a linear model where the value of y is determined as a sum of different effects. Firstly, there is some constant or intercept, and then the effect of the predictor or independent variable x is at the top, and beta is this effect. It's called the coefficient of regression. And finally, the third effect is random residual noise. The estimation of parameters, namely the intercept and coefficient of regression, is done in such a way that the predicted value of the outcome is as close as possible to the observed value. In a univariate case, the estimates of beta can be obtained with these simple, relatively simple formulas. That is, the estimate of beta is the covariance of x and y divided by the variance of x. Both intercept and regression coefficient in linear model have very clear physical interpretation. The intercept mu is expected value of y if the the value of predictor x is 0, and coefficient of regression beta tells how much y change when x is changed by single unit. So let us consider an example with linear regression model, and let's be practical. So we are going to predict height based on sex. So the sex is coded as 0, 1, 0 for female and 1 for man. And then on this graph every dot represents a person with particular sex and height. And in the dataset depicted at the picture, it contains 48 males, so there are 48 dots on the right side, and 52 females. The following estimates are obtained. The intercept is 167.6 and the estimate of beta is 12.6. My question is, can you interpret these values? What is this 167.6 and what is this 12.6? What is the physical meaning of this? The interpretation of the intercept means that when x is 0, this is the expected value of the outcome. In other words, the expected height of females is 167.6 centimeters. And the beta is telling how much x changes, how much y changes when x changes by 1. In other words, this is the expected difference between the height of male and female. Or you can tell it the other way around. You can tell that height of males is the intercept plus the regression coefficient, which is 180.2 centimeters. Now, important point is that the regression coefficients are scale dependent. So let height is measured in millimeters instead of centimeters, and then the estimates which would have been obtained on exactly the same data would be the intercept is 1676 and the coefficient of regression would be 126. So the measuring height in millimeters instead of centimeters is exactly the same as multiplying all the outcomes by 10. So this would multiply all the estimates by 10, as you can see. But the data are exactly the same. So now we are more equipped to answer the question for the panels A, B and C, where association is stronger. So would you still guess that for this data the strongest association is for A, and then it's less strong for B and less strong for C, when measured with the coefficient of regression? We may want to take into account the fact that with regression coefficients may be not the best measure to characterize the strength of association because it is scale dependent. So actually, if you run regression analysis on this data, you would figure out that the coefficient regression for the data presented on panel A is 0.95, while the coefficient regression for the panel B is bigger, and for panel C it's much smaller. Still, intuitively looking at this data, I would say that association on A is stronger in the sense that the values of x and y are going very neatly together, and then I can also see the dependency between x and y on panel B, and I can hardly see it on panel C. So in this way, the coefficient regression is a little bit counterintuitive measure association, so it's perfectly valid and makes a lot of sense when we need clear physical interpretation of our results. So the problem with these data sets was that they were scaled in different manner, and now if I correct the scale for all these data sets A, B and C, so I unify the scale, I can better see why the coefficients of regressions are so different between these data sets. Now, the intuitive measure of regression would correspond to a feeling how neatly x and y go together. But in this case, of course, we need something scale independent. Our observation now is that the regression coefficient changes with scale. The larger is the variation of the outcome, the larger is the coefficient. We can estimate how much y changes when x is changed by 1 unit with coefficient of regression of y onto x, but we can also estimate how much x changes when I is changed by 1 unit by this formula. So the idea would be that scale-independent measure of association should contain both the variance of x and the variance of y in the denominator. And by using this idea, you can come to the coefficient of correlation, Pearson coefficient of correlation, which is defined as the covariance between x and y divided by the square root of the product of variances of x and y. So the coefficient correlation is constrained between -1 and 1, and when this coefficient is 1, there is a perfect linear dependency. As x increases, y also increases. When coefficient of correlation is -1, there is a perfect correlation, but now reciprocal. So as x increases, y decreases. While when coefficient of correlation is 0, there is no, at least linear, relation between these two variables. The square of the coefficient of correlation, also called coefficient of determination, gives the proportion of variance explained, and this is a very important statistical measure of the strength of relation between two variables. And of course it varies between 0 and 1, with 1 being a perfect correlation, either -1 or +1, but this means we can perfectly predict x from y or y from x. And when coefficient of determination is 0, we don't see any linear dependency between x and y. So here are a few examples of correlations, and so let's look at the upper panel. On the left hand side, there is a perfect correlation between two variables, and the coefficient of correlation would be 1, and then this correlation becomes less and less perfect, and the coefficient correlation becomes 1 when there is no relation between two variables. When the relation between two variables becomes reciprocal, then the coefficient of correlation becomes negative. Now, note that coefficient of correlation is scale independent, and therefore when we see a perfect linear correlation, this is in the down panel, it doesn't matter what is the angle of this relation, what is coefficient of regression. The coefficient of correlation would still be 1 or -1 when the relation is reciprocal. And here is interesting point. You see there is no any number here, and I would like to ask you why there is no number here. Well, the answer is that coefficient of correlation is defined as covariance between two variables divided by square root of variance of both of them, and then for the y random variable there is no variation. it's simply constant. And then the variance of y becomes 0, and you get in your formula division by 0. Therefore, this number is simply not defined. You're trying to compute correlation between a random variable on x and a constant, and this is not something which makes any statistical sense. So let's look at correlation for our data. As you remember, with coefficient of regression, the strongest regression was for data presented on panel B, and then for A, and then on C. Now if you look into correlation, then for data presented on panel A, the coefficient correlation is almost 1. It's so close to 1 that I had to round it up in order to avoid very long number. And then for panel B, the coefficient of correlation is 0.5, and for panel C is this 0.27. So indeed, A is greater, B greater C, and the coefficient correlation indeed corresponds to this intuitive feeling of how neatly two random variables go together. So let's try to think of yet another aspect of association. Here I have the data. It's different data from previous slide, and for all these data sets A, B and C, I have roughly the same coefficients of regression and coefficients of correlation. They are not exactly the same, but quite roughly they are the same. So let's assume they are the same. But would that mean that the strength of association in all these three panels is more or less equal? So to answer this question, we need to first look at what is changing between A, B and C, and we can figure out that there are only 10 observations in panel A, 30 observations in panel B, and 70 observations in panel C. So while the magnitude of association is measured with coefficient of regression and correlation coefficient stays more or less the same, the amount of data points, the amount of evidence that this correlation, this association is true is different. And given the same amount of association, of course the experiments with more observations provide more evidence. The observed association is likely to appear by chance. So, yet another way to characterize association is to ask the question, what is the chance to observe this strong or even stronger relation by pure chance? And this chance is termed p-value. The lower is p-value, the less likely is association to appear by pure chance. Consequently, the statistical significance measuring our confidence in our belief into the fact that this is true association becomes higher. To obtain p-value, we can use the score test, and we will use it later on quite a bit. This score test is defined as, we will call it t-square, as the coefficient of determination. You remember this is square of the correlation coefficient between x and y multiplied by the sample size n. Under the null hypothesis of no association, this test is distributed as chi-square with 1 degree of freedom. And for this distribution, you know many things. For example, you know that if you obtain the value of t-squared more than 3.84, you can say that p-value is less than 5%. So nominally, you would consider this significant result. Okay, let's revisit our results for these data sets where we have seen roughly the same coefficients of correlation and regression, but the amount of evidence, the number of observations was different between these three data sets. And then you can see that with panel A, where we have small number of observations, the p-value is 1.7%. For panel B, where we have 3 times more observation, the p-value becomes 4 by 10 to the power of minus 5, and then for panel C, it becomes really significant, incredibly significant actually, with p-value close to the 10 to the minus 12. How do we compute these p-values? Okay, first we can of course need to compute the coefficients of determination. So we take the square of the coefficient of correlation, and they would be approximately the same again, and they are listed here, 0.53, 0.45 and 0.53 again. Now you need to multiply these coefficients of determination by the sample size, and then you would obtain the score test statistic values. And from these values you can easily compute the p-values. So if we go back to our data and try to answer where association is stronger, this is really very poorly defined question, because you can measure association in many different ways. If you use regression coefficient, then panel B is the champion, because as x1 changes, y1 changes a lot, by 3.3. But if you use correlation, which measures how neatly the x and y go together, then the panel A, predictor X, is the champion. While when you use statistical strength, p-value, then the data presented on panel C are statistically more strong, just because there is a huge number of data points there. So the point to take home is that there are several ways to measure associations, and these ways are complementary. Regression coefficient is a very useful measure which has clear physical interpretation and allows easy prediction. This coefficient is dependent on the scale of outcome and predictor though. Coefficient of correlation and determination provide a pilling measure of how neatly the outcome and the predictor go together, and how visible is this association. P-value tells you how much evidence are provided by the data to rule out the hypothesis of association. These 3 measures of associations are complementing each other. Now, something which is really worth to mention when you're talking about linear regression, you do assume linear dependency between outcome and predictor. If you look at the lower panel in this graph, you can see that there is a strong relation between x and y. There is a clear shape. At the same time, if you try to do linear regression or compute correlation and then p-value, then you will find there is no correlation whatsoever, and the coefficient of regression is 0 for all the data presented at the lower panel, just because a linear line would always be very flat. These data are balanced along the y-axis. So let us look what let us now use this knowledge to consider genetic data analysis. So when you study genetic data, you are interested in the relation between some outcome y, which could be some trait, height, weight, and a genetic predictor G. So let G is a single nucleotide polymorphism with 2 alleles A and B, and then 3 genotypes are possible. We can formalize different genetic models by coding this predictor variable G in different ways. So we are going to formulate simple single regression coefficient model where G is coded according to different models. So first of all, the additive or B allele dose model would assume the following coding. So you say this is B allele dose, and in AA genotype there are no B alleles, so the dose of B is 0. In AB the dose of B is 1, and in BB the dose of B is 2. Then you could assume dominant B model. Then you say that it doesn't really matter whether B is heterozygote or in homozygote, the effect is the same. And then you would code AA with 0 and AB and BB with 1. Then you could consider recessive B model. You say that the effect of B is only visible when B is in homozygote. Then you would code AA and AB with 0 and BB with 1. And finally there is a somewhat exotic over-dominant or heterozygous model where you say that both homozygotes have the same effect while heterozygote might have a different effect. And then you code AA and BB with 1, with 0, and AB with 1. But we sometimes would like to allow for differential effects for all three genotypes. And then you can do that by formulating two predictors, G1 and G2. These G1 and G2 can be defined in a number of ways, for example via G1 coded as additive effect and G2 coded as over-dominance effect. In this case, beta 1 would give you additive effect of allele B, while beta 2 will estimate dominance deviation. And this model is tested against the null model, where no genotype is present, resulting in 2 degrees of freedom test. So chi-square, you would compare it to chi-square with 2 degrees of freedom now. In general, genetic association analysis is done using standard statistical methods, but there is specifics of this analysis, and this specific comes from the special nature of the independent variable of interest, being the genotype. And this genotype is a real object for particular genetic laws.